Welcome back, students. In this section, I'll be talking to you about protein metabolism. So, are proteins used for energy? Are proteins being degraded and help us to get ATP? Can we do this? Usually not. But in certain, still, are the proteins being metabolized? Yes, the proteins have a constant turnover. Some proteins are constantly being synthesized in our body. Some proteins are constantly being degraded in our body. So first, before I go to the individual amino acids and their metabolisms and their functions, let me just tell you what is happening within the body as far as proteins are concerned. Now, proteins are mainly structural. They have lots of functions within our body, but usually not serves as a source of energy except during starvation. And during starvation, the muscle proteins are also broken down to give us energy, to give us glucose. But the body proteins are undergoing a constant turnover. So, how much is the half-life? How will you determine whether a protein is going to die within 3 minutes, within 2 minutes, within few seconds, or within so many years, months? How is it all possible? So proteins have a definite half-life, and this half-life varies from some proteins from minutes to some proteins for years together. So when a protein degrades, it releases its amino acids, and these amino acids are used for the generation of a new protein or for the sake of energy, depending upon the energy status of the body. If the energy status of the body is good, the new amino acids are used for the synthesis of a new protein. Remember, the body does not have appreciable stores of amino acids. Now, again, as I was telling, this protein is undergoing a constant turnover. Around 300 to 400 grams of protein is being synthesized per day and being degraded at the same time. So in the body we have two major systems by which these proteins are degraded. One is energy dependent, the other is non-energy dependent. The energy dependent mechanism by which proteins are degraded is called as the ubiquitin dependent mechanism for the degradation of proteins. Now, this process, ubiquitin proteasome proteolysis. So in this ubiquitin molecule is used and energy is used, and the proteins are undergoing a degradation. So we have a protein here. This protein, what has happened to it? Some problem is there with the protein, or it has reached the end of its life. Now is the time to degrade this protein. It is time to bring about its proteolysis. So at that time, the body has certain ways by which it starts attaching ubiquitin molecules to this protein. Remember, if it is ubiquitin molecules are present to this protein, you call it a ubiquitinated protein complex. Now, once it is ubiquitinated, once ubiquitin has been added to this protein, it will be recognized. Recognized by what? It will be recognized by another proteasome complex. This is a macromolecular protein complex. Okay, it is mainly like a barrel shaped. This is like a barrel shaped macromolecular complex containing lot of other proteins. Now, this barrel will recognize this ubiquitin protein, and this ubiquitinated protein will enter into this barrel. Once it comes out, it will be all fragmented and proteolysis will have taken place. At that time, the ubiquitin is released, and this ubiquitin molecules are reutilized for ubiquitination of another protein molecule. So this process by which a ubiquitin is added and it is recognized by a proteasome is energy dependent, because for the sake of attaching ubiquitin we require ATP. So this is an energy dependent mechanism. Once the amino acids or the proteolytic fragments are there, they are further converted into other different amino acids. The individual amino acids will contribute to the amino acid pool of the body. Now, what determines whether a ubiquitin is going to be attached to a protein or not. So there must be some mechanism in the body by which the body will come to know, okay, this is the time for this protein to die, or this is the time for this protein is not proper, so this protein has to die. So it might be a misfolded protein, or it might be the half-life has been reached and now is the time to degrade. So what are the sequences which help the ubiquitin to recognize this protein? And the N-terminal amino acid sequence tells the ubiquitin what to do with it and when to do ubiquitination of a particular protein. So let us look here. This ubiquitin proteasome proteolysis is mainly for endogenous proteins, that is the proteins which have misfolded or have reached the end of their life. So first, proteins are covalently attached to ubiquitin. This is called as ubiquitination. This is an energy dependent process. It is a 3-step process and a polyubiquitin chain is attached to the protein. Now, during this attachment, the first bond is formed between the glycine of ubiquitin and lysine of the protein. Now, what this ubiquitinated protein is recognized by proteasome, a barrel-shaped macromolecular proteolytic complex, and protein degraded and ubiquitin reutilized. Ubiquitin is what is ubiquitin? Ubiquitin means ubiquitous in nature, present in all the cells. Hence the word ubiquitin. It is a small globular protein by itself. Now, half-life of proteins can vary from minutes to years. If you look at a regulatory protein where its levels have to be regulated, minute to minute, second to second, they have very short half-life. Because minute its action is over, these regulatory proteins have to be degraded. If it is a misfolded protein, yes, again the half-life is very short. So long-lived proteins, can you give an example? We have collagen, the structural protein, which has a very long half-life. So long-lived proteins can have as many as days to weeks, and majority of the body proteins are having a long-lived protein. Majority of the body proteins are long-lived proteins. Structural protein, collagen, has months to years, very long-lived proteins. So the half-life is influenced by N-terminal residue. As I said, if it is a N-terminal residue is serine, the half-life is around 20 hours, whereas if the N-terminal residue is aspartate, it is around 3 minutes. Plus, we have a certain sequence present on the protein for which we have very short half-lives. These are called as the PEST sequences. PEST sequences. PEST stands for P for proline, E for glutamate, the one letter abbreviation of glutamate is E, S for serine, and T for threonine. So a protein which is having PEST sequence will have a very short half-life and will be degraded faster. So this is one mechanism by which the body degrades proteins and results in the formation of amino acids. Remember, this is mainly energy dependent. Do we have any other mechanism? Yes, these mechanisms are energy independent and are mainly used for exogenous proteins. So proteins which are come to the body from some other source. So this is done by lysosomes. Any cell which is having lysosome can do lysosomal protein degradation. For this, the protein is taken up by endocytosis, will form a phagosome, then a phagolysosome, and the lysosomal enzymes will bring about proteolytic degradation. So these are this is a non-energy dependent protein degradation by lysosomal enzymes, which are acid hydrolases, and mainly for extracellular proteins which are taken up by receptor mediated endocytosis. So now all this has taken place and we have got what is called as the body amino acid pool is there. So what are the contributors of this amino acid pool and what is the fate of this amino acids? So let us look at it. Amino acid pool is contributed by body protein degradation. Dietary proteins also contributed. Synthesis of non-essential amino acids also contribute, and they are used for body protein synthesis of biologically important compounds like creatine, neurotransmitters, purines, pyrimidines, and other nitrogen-containing compounds are synthesized from these amino acids, or these amino acids can also be catabolized to produce energy. So let us look at the catabolism of different amino acids. Now, what is a characteristic feature of any amino acid? As I said, they have to have a amino group and the carbon skeleton and the carboxylic acid. Now, the metabolism of the amino group and the carbon skeleton is slightly different. This amino group, amino acids are very specific, are very special because they have the nitrogen containing compounds, and the rest of it is carbon. So its metabolism is different. The metabolism of the amino group is different. This amino group is usually cleaved off or is removed from the amino acid and results in the formation of urea, whereas this carbon skeleton can have different fate depending on the type of amino acid, whether it is glucogenic or ketogenic. If it is glucogenic, this carbon skeleton, what it will do? It will produce glucose. If it is ketogenic, it will produce ketone bodies. So with this, I'll just give you a schematic diagram of the amino acid general metabolism. As I said, amino group and carbon skeleton. Look at this slide. The amino group can go and form urea, and urea is excreted out into the urine. The carbon skeleton can form glucose in glucogenic amino acids and ketone bodies in ketogenic amino acids. Remember, all amino acids are glucogenic except leucine and lysine. The ketogenic amino acid include leucine, lysine and the PITT amino acid, that is phenylalanine, isoleucine, tyrosine and tryptophan. These are the ketogenic amino acids. So let us look at any other thing that is possible from this carbon skeleton. Yes, carbon skeleton can also liberate carbon dioxide during these processes. Now, other than this, let's look at first how urea is going to form. My focus of this section will be on how this amino group is removed and how it forms urea. So, how is this amino group removed from different amino acids? What removes it? How is it removed? So, we call it as removal of nitrogen from amino acids. Now, there are two processes that are important in this. One is called as transamination and the second is called as oxidative deamination. What is transamination? Transamination, the name itself tells you that transfer of amino group. So, in transamination, there is transfer of amino group. From what to what? Most of the time, suppose this is an amino acid, it transfers its amino groups. Once it transfers, what happens to an amino acid? It gets converted to its respective keto acid. Okay. Now, it has to give its amino group to something. To what does it give? It gives its amino group to another keto acid, and this usually is alpha ketoglutarate. Where have you heard of alpha ketoglutarate? This is an intermediate of the TCA cycle. So alpha keto acids, remember, alpha ketoglutarate is a keto acid which takes up the amino group from any amino acid, and this gets converted to glutamate. Alpha ketoglutarate gets converted to glutamate. Glutamate is an amino acid. This type of reactions in which an amino group is transferred is called as transamination, or amino and the enzymes which catalyze these are called as amino transferases. They have transferred amino group. And what else is required for this type of reaction? This is a familiar reaction. What else is required? Pyridoxal phosphate. So PLP plays a central role in amino acid metabolism. By this mechanism, all the body amino acids, all the different amino acids which are present in the body can transfer their amino group to glutamate. Now what? Glutamate can be transported to the site where urea is going to be synthesized, where from glutamate we can get out urea. So this process by which later on I will talk to you what happens to glutamate. So let us first concentrate on this transamination process. So as I said, removal of nitrogen from amino acid requires transamination and oxidative deamination. What is transamination? Transfer of alpha amino group. Amino transferases catalyze it and require PLP. Remember, this reaction is reversible. So if this reaction is reversible, what will happen? What will happen? What determines whether this will go like this or this will go like this? In which direction it goes depends upon the energy status of the cell, depends upon the amino acid requirement by the cell. If a particular amino acid is required, suppose there is translation, suppose that cell is undergoing actively synthesizing proteins. If it is, that is translation, and during translation all the 20 amino acids have to be present within the cell. One amino acid is not there, and this is a non-essential amino acid. Then transamination can occur in the reverse direction, and it can help in the synthesis of this amino acid, and the protein synthesis will continue. This is the importance of this transamination. But suppose the protein is in a well-fed state and there is no protein catabolism as such is taking place, and there is no protein synthesis, and this protein is going to be degraded and used for energy, then it goes in this direction and all the amino groups are transferred to glutamate. So transamination reactions are reversible, and transaminases are specific for the amino acid 1, the first amino acid, and usually the acceptor is alpha ketoglutarate. So I'll give you an example how it is being done. This is a schematic diagram of how action of amino transferases takes place. Remember that these amino transferases are specific for this amino acid. So amino groups from various amino acids are funneled into glutamate, and this can also help in the synthesis of non-essential amino acid as the body requires. Suppose the cell is in an active translating mechanism, then it will require all the 20 amino acids. At that time the non-essential amino acids are synthesized. Now there are certain exceptions to everything. I said all the amino acids can give their amino groups to glutamate, except this is again an MCQ question, which are the amino acids which do not undergo transamination. They include lysine, threonine, proline and hydroxyproline. These 4 amino acids do not undergo transamination. Another few example important which are clinically important also, alanine transaminase, where alanine gives its amino group to alpha ketoglutarate and alanine is converted to pyruvate and glutamate is formed. Another action is aspartate transaminase, where aspartate is converted to oxaloacetate. Now these two reactions which are catalyzed by alanine transaminase and aspartate transaminase are something which you cannot forget. These transaminases are central to a lot of reactions, form a central role within the body, and they are the ones which are used as liver function tests also. In clinical enzymology, in liver function tests, in every condition, gluconeogenesis, everywhere, these amino transferases form a key role. So let's go ahead. What happens to this glutamate? What is the fate of this glutamate? What else can happen to it? So this glutamate will finally reach the liver. In the liver, it undergoes what is called as oxidative deamination. So you have got one term, that is transamination. Now I go ahead with another term, that is oxidative deamination. So all amino acids have given their amino groups to glutamate. Glutamate has reached the liver. What happens to glutamate? It is acted upon by an enzyme called as glutamate dehydrogenase. Now remember, we can use GDH as a short form. Pyruvate dehydrogenase becomes PDH. So like that, glutamate dehydrogenase can also be used. What does the enzyme name mean to you? And what is the type of reaction that is catalyzed? It is oxidative deamination. So what is required? It requires oxygen. Oxidative deamination means that it requires oxygen, but instead of oxygen, what is used here are, it undergoes oxidation by giving it to NAD plus, is converted into NADH plus H plus. Has it undergone oxidation? Yes. And during this process, deamination, amino group is released as ammonia. And what happens to glutamate? If glutamate loses its amino group, it gets converted to any amino acid. When it loses its amino group, gets converted to its respective keto acid. What is the keto acid of glutamate? It is alpha ketoglutarate. glutarate, which is an intermediate of the TCA cycle. So let us look at this reaction. Where is it occurring? This is occurring in the liver and kidney. It is occurring in the mitochondria. That is important, again MCQ points of view. This results in the liberation of free ammonia along with simultaneous oxidation. Glutamate is the amino acid. Now I have got one more terminology here, that is transdeamination. So transamination followed by oxidative deamination, you call it transdeamination. I'll tell you briefly more about glutamate dehydrogenase. What is this? Remember, this is one of the special enzymes within the body which can use either NAD or NADP. It can use either NAD or NADP. This reaction is reversible. This is one of the few reactions in the body which are reversible. So this is oxidative, especially oxidative deamination. This reaction is reversible and it can use both NAD or NADP as the coenzyme. During the reverse reaction, that is during liberation of ammonia, NAD is used, and during reductive amination, NADPH is used. When it is going in this direction, NADPH. When it is coming in this direction, NAD is used. So direction of the reaction, what determines when this is formed or when this is formed? It depends upon the relative concentration of glutamate, alpha ketoglutarate, ammonia, and the redox state of the cell. So depending on all this, this direction will vary. So GDH is both anabolic and catabolic and can be used for the synthesis of non-essential. So if it is catabolic, it goes further, so it is catabolic. At the same time, if it goes in this direction, it is anabolic, and so it can be used for the synthesis of non-essential amino acids. Now this is also one more special enzyme which is zinc containing, present in the mitochondria. Its inhibitor include ATP and GTP, which are the allosteric inhibitors, and ADP and GTP are its activators. So when energy levels are low in the cell, energy is not there, energy levels are low. When is it low? ADP is low and ATP is low. If energy levels are low means ATP is low, then amino acid degradation is going to be higher. So during this time, this will take place. So let us go further in the next section where I will be talking about other different mechanisms by which oxidative deamination can take place. Thank you.